Hello, I am Dr. Aizaz from MedicoVisual.com, and in today's visual lecture we will talk about the formation of body cavities. And by body cavities I mean the formation of peritoneal cavity, the pericardial cavity and the pleural cavity. And along with that, in the later lectures we will also talk about the formation of diaphragm. So let's start the video from the very basic concept that let's suppose here is the embryo which was embedded into the uterus of its mother, and here you can see this appearance, this rough appearance of this embryo is due to the presence of villi that are formed by the trophoblast. Especially from outside, this is the syncytial trophoblast, and inner to that will be the cytotrophoblast. Now if we take a knife and if we cut it like this, if we remove this trophoblast and if we cut and remove this trophoblast, what you will see is that here is this extraembryonic somatic mesoderm. So inner to the syncytio and cytotrophoblast, inner side is underlined by the extraembryonic somatic mesoderm. And if you don't know about these structures, please watch my lectures on second and third week of development of embryology. Now if we take a sharp object and if we cut a section of this embryo further, here you can see that these things will be seen. Now what is this? Let me label these structures for you. Here is the ectoderm and here is the endoderm. So between ecto and endoderm, here is the intraembryonic mesoderm. Here also intraembryonic mesoderm, and of course there would be the notochord, and above that the neural tube will be formed, but I haven't shown those structures. So here is the ectoderm, and here you can see this ectoderm, it is continuous at its periphery, it is continuous with the membrane of amniotic cavity or amnion. So basically this ectoderm, it is surrounded by the amniotic cavity, and it is continuous with the amniotic cavity membrane or amnion. Similar to that, here is the endoderm, and this endoderm, it is surrounded by another cavity called yolk sac cavity, and it is continuous with the membrane of this yolk sac cavity. So this is the yolk sac cavity membrane. It is also called yolk sac endoderm. And here please remember that here is the amniotic cavity membrane. So ectoderm is continuous with the amniotic cavity membrane, and endoderm is continuous with the yolk sac cavity membrane, and between ecto and endoderm is the intraembryonic mesoderm. And this intraembryonic mesoderm is continuous laterally with all this extra amount of mesoderm which is not within the embryo proper, and this is the extraembryonic mesoderm. So this is the intraembryonic mesoderm. This intraembryonic mesoderm is laterally continuous with the extraembryonic mesoderm. So all these things we have studied already in the previous lecture, so it is a bit of revision. Now what actually happens during the course of development, small spaces appear within the extraembryonic mesoderm, and later such small spaces, they also appear within the intraembryonic mesoderm as well. So first small spaces arise within the extraembryonic mesoderm, and later small spaces, they also arise within the intraembryonic mesoderm. The small spaces that appear within the extraembryonic mesoderm, they coalesce to form the extraembryonic celom. And similar to that, the spaces that arise within the intraembryonic mesoderm, they coalesce to form the intraembryonic celom. So here you can see that this is the intraembryonic celom. So intraembryonic celom, it is laterally continuous at its periphery. It is continuous with the extraembryonic celom, except at the site of septum transversum that we will discuss later. So now you should be very clear about these structures. But one thing you must notice here, that here is this something where there is no cavity. Here no small spaces appear in this region, and this mesoderm, it remains intact and it forms the connecting stalk. So this connecting stalk, it connects the extraembryonic somatic mesoderm here with this embryo like this. Now, what are the structures that you can see? Also appreciate that the mesoderm, it is not a homogeneous structure. It has been divided into different parts. If I label all these structures, here is the paraxial mesoderm, here also paraxial mesoderm, and then there is the intermediate mesoderm. It is continuous with the intermediate mesoderm. And then here is the intraembryonic somatic mesoderm, intraembryonic somatopleuric mesoderm, and here is the intraembryonic subplanknopleuric mesoderm. Here also intraembryonic somatopleuric mesoderm and intraembryonic subplanknopleuric mesoderm. Now, intraembryonic somatopleuric mesoderm is continuous with the extraembryonic somatopleuric. This is the extraembryonic somatopleuric mesoderm, and here is the intraembryonic subplanknic mesoderm. It is also continuous with the extraembryonic subplanknic mesoderm. Here as well, intraembryonic subplanknic mesoderm continuous with the extraembryonic subplanknic mesoderm. Here as well, intraembryonic somatic mesoderm is continuous with the extraembryonic somatic mesoderm. And of course, this is also the extraembryonic somatic mesoderm. So extraembryonic somatic mesoderm is at two places. At one place, it is covering from outside the amniotic cavity, remember, and at the other place it is underlining the cytotrophoblast. Here is also extraembryonic somatic mesoderm, here is also extraembryonic somatic mesoderm, and here this one is the extraembryonic subplanknic mesoderm. So please keep that in your mind. Now let's remove this extraembryonic somatic mesoderm. So here we will see that only embryo is left. And now let's uncut it like this. And here there is the complete embryo. Now if I want to take a longitudinal section of this embryo, what I mean by longitudinal section is that, let's suppose here we cut it like this. If we cut it like this would be the longitudinal section of this embryo. So let's take a longitudinal section of this embryo. So here is the longitudinal section of the embryo, and here again you can see all these structures: the paraxial mesoderm, intermediate mesoderm, and intraembryonic, which layer? Intraembryonic somatic layer and intraembryonic subplanknic layer, and here is the septum transversum. We have already discussed about the septum transversum, that septum transversum is basically a block of tissue, and at the site of septum transversum you can see that intraembryonic celom, it is not continuous with the extraembryonic celom, because here this block of tissue is present. Now let's talk about the craniocaudal folding. So here again, here is this the neural tube that I have shown here, and there is that paraxial mesoderm, intermediate mesoderm, and the somatic and subplanknic layer of intraembryonic mesoderm, which is continuous with the extraembryonic mesoderm. And by the way, just remember that between the intraembryonic somatic and subplanknic layer, here is this space, this cavity called intraembryonic celom, and it is at its periphery, it is continuous with the extraembryonic celom. Now in craniocaudal folding, what happens that due to the development of this neural tube, from here the embryo will be folded, and at the tail end also the embryo will be folded ventrally, or in this diagram you can say downward, but it is not actually downward, it is ventrally. So let's see the animation. So here you can see the craniocaudal folding has occurred, and as a result of craniocaudal folding, this here you can see here is this intraembryonic celom. So one part of intraembryonic celom is here, and the other part of intraembryonic celom is here. We will discuss this in a bit more detail later, but also please note that a part of endoderm, let's see it again. So here you can see that this part of endoderm along with the yolk sac cavity, it is embedded here. This is the foregut, and here also this part is embedded. This is the hindgut, and between that is the midgut, and it is still continuous with the with this yolk sac cavity and through this duct called omphaloenteric duct. We have already discussed these things in the GIT embryology course. So this is the craniocaudal folding, but what about the transverse folding? So now let's briefly also talk about the transverse folding. So now what we will do is that we will uncut this, we will uncut whole of this, and we will take a transverse section of it. What I mean by transverse section, that let's suppose we take a section from here like this. So if we uncut it and if we take a transverse section, then you can appreciate the transverse folding clearly. Now if you focus here at this transverse section, here is this tail end, and you can see what happens in the transverse folding, that the embryo, it rotates from here like this. So it rotates from here like this, and here it will be pinched inward like this. So let's watch the animation and things will become pretty much clear. So here you can see what is happening here. So this is the transverse folding. So as a result of transverse folding, you can see here, I am right now focusing mainly on the body cavities, the intraembryonic celom. So here you can see this part of intraembryonic celom, it is formed here, and it is now surrounding the this GIT tube. So here you can see it is surrounding the GI tube. Here also it is surrounding the GI tube. So as a result of transverse folding, here you can see what happened to this intraembryonic celom. So intraembryonic celom has achieved a particular shape, it has attained a particular shape, and it is surrounding this primitive GI tube. So here also you can see it is surrounding the primitive GI tube. So that is what exactly happens as a result of transverse folding. Now let's see the final image of the embryo after the craniocaudal and transverse folding. Now this is the situation of embryo after the craniocaudal and transverse folding. Now here you can see one layer of mesoderm. It is surrounding the yolk sac, I mean the primitive GI tube. Here you can see it is surrounding the primitive GI tube. This layer is the intraembryonic subplanknopleuric mesoderm, and it is continuous with the extraembryonic subplanknopleuric mesoderm. And here this layer from inside, it is lining the ectoderm. So here this is the intraembryonic somatic mesoderm. Actually ectoderm is going to form the body wall. So this layer, it is underlining the body wall. That is why it is called intraembryonic somatopleuric mesoderm, and it is continuous with the extraembryonic somatic mesoderm. And between these two is the intraembryonic celom. And this intraembryonic celom is basically the primordium of all three body cavities. Please keep that in your mind. Now what I will do is that I will remove each and every distraction, and I will only show you the intraembryonic celom along with the intraembryonic somatic mesoderm and subplanknic mesoderm. So here is the color-coded mesoderm, intraembryonic mesoderm, the paraxial mesoderm, then there is the intermediate mesoderm, and this purplish one is the intraembryonic somatic mesoderm, and this greenish one is the intraembryonic subplanknic mesoderm. And between these two layers is the between these two layers is the intraembryonic celom, and here you can see this block of tissue is the septum transversum. Now what actually happens that as a result of craniocaudal and transverse folding, let's see what happens. So here is this U-shaped intraembryonic celom. So craniocaudal folding, and then there is transverse folding. Actually please remember that all these three foldings, they are occurring simultaneously, but for the sake of ease of explanation, I am showing you one by one. I will assume in this animation that craniocaudal folding occurs before the transverse folding, but please keep that in mind that actually they occur simultaneously. So let's watch the animation, and here you can see folding is going on, and as a result of transverse folding, now what you can see here, that these walls, they are coming close together. So this is the intraembryonic somatic mesoderm. Now here what will happen that these two layers of intraembryonic somatic mesoderm, they will become stitched together like this. And what about the intraembryonic subplanchnic mesoderm? They will also become stitched together. Let's see how. So here again inside you can see here is the intraembryonic subplanchnic mesoderm. These two layers, they will also become fused together like this. Let me show you exactly how it happens. So let's move forward. In the next visual illustration you can see how they fuse together. Let's see it again. So before the fusion it was like this, and after this fusion it has become like this. So now inside them is the intraembryonic celom. Now intraembryonic celom, and what about this? This huge space here? Actually, you know what? Here is the GIT, primitive GI tube, and it has three parts: the foregut, midgut, and hindgut. And the midgut, it is actually continuous with the through umbilicoenteric duct with the yolk sac cavity. If you don't understand these things, please watch our video on gastrointestinal embryology. We have a complete 3D course on gastrointestinal embryology. Please do check out on medicovisual.com. So due to presence of this huge duct at this time, it cannot be stitched together here. So here actually what happens that if I show you the complete cut section, so here this intraembryonic subplanchnic mesoderm, it is continuous, and it is continuous with the covering of yolk sac cavity, and that is the extraembryonic subplanchnic mesoderm. So here the intraembryonic subplanchnic mesoderm is continuous with the extraembryonic subplanchnic mesoderm, and also here you can see intraembryonic somatic mesoderm. Here it is continuous with the extraembryonic somatic mesoderm, and please remember here there would be that connecting stalk which I haven't shown in this 3D diagram. Now here you can see that actually we have, after the folding and stitching process, we have lost the connection of intraembryonic celom with the extraembryonic celom at most of the places except near this midgut region, except near this region where this umbilicoenteric duct or vitelline duct is present. Here there is still the connection between the intraembryonic celom and extraembryonic celom. So let me show you exactly where is the connection. So here you can see this is the whole, this is the extraembryonic. So this is basically the extraembryonic celom. So here you can see if we go from extraembryonic celom, we can go inside the intraembryonic celom at this region. Here also we can go. So near this midgut region, there is still a connection between the intraembryonic and extraembryonic celom. Now please understand that if we go back, here you can see that when it was not stitched together, here there was a connection between extraembryonic celom. You remember that whole this is from outside surrounded by the extraembryonic celom. So earlier there was that connection of extraembryonic celom with the intraembryonic celom at most of the places. But after the conclusion of this folding process and this fusion process, now this connection, it has remained only at this small place near the midgut region. Also please note that before the folding process, the septum transversum, it was the cranial most structure, but as a result of folding it has came here, and here it will form the diaphragm. Later we will see that how it forms the diaphragm, and it has some other functions as well that we will not go into detail right now. Now here is the longitudinal section of the embryo, and here you can see this is the ectoderm, here is the neural tube, and here is this body cavity, the intraembryonic celom. And here you can see this connection has now been narrowed down. Initially there was that wide connection. Let me show you exactly what I mean. So initially there was that wide connection between the primitive GIT and the yolk sac cavity, but but this connection ultimately it is pinched off and it becomes very narrow. Here you can see that it becomes narrow, and ultimately after the herniation process and midgut looping and all these things that we have already discussed, after those process, when the herniation is concluded and the GIT loops as they get back into the abdomen, ultimately this connection is lost. Now here is that same primitive GI tube, and here is the intraembryonic celom. And what happens to this primitive GI tube, that it will move slightly inward like this, and as a result of this a mesentery will be formed, and we will not talk about the mesentery in detail, but what I will do here is that I will take a section of this section of this intraembryonic celom, this primitive intraembryonic celom. And by the way, here is the mesentery. So as it is pushed inside, as this primitive GI tube is pushed inside the peritoneal cavity, well, it is primordial peritoneal cavity, actually it is right now the intraembryonic celom, and it will form the peritoneal cavity. So it is pushed into the peritoneal cavity. So here you can see a double layer is formed, and this double layer is the dorsal mesentery, and here is that block of tissue called septum transversum. Now what happens that the septum transversum, it will also grow laterally like this, and as a result the two canals are formed around this the primitive GI tube and mesentery. Two canals are formed that we will just talk about. But before that, for the sake of simplification, because this is kind of distracting me, let's remove this this connection and let's make it simple. So here let's make it simple, and now let's see what happens. I think let's remove everything else and let's just talk about this body cavity. So here is that complete primitive body cavity, and here within that is the septum transversum. So here is the septum transversum, and here you can see here is the septum transversum. Now this layer which was surrounding the primitive GI tube, let me show you exactly. So here is that layer which is surrounding the primitive GI tube. This is going to form the visceral peritoneum. It is going to form the visceral peritoneum because it is surrounding the viscera, and it is, by the way, intraembryonic subplanchnic mesoderm. It is visceral layer or subplanchnic layer of the peritoneum, and which the other layer which is surrounding the body wall, it is the parietal layer of peritoneum. It is the somatic mesoderm, and it is the parietal layer. Here also it is the parietal layer of peritoneum. Now to make it easier for you to understand, I will take a longitudinal cut section of this body cavity. So here is the longitudinal cut section of body cavity, and here you can see. Now let's talk about what structures will form all these things. Now here one thing, please note that this septum transversum, this block of tissue, initially it lies in the cervical region, but as the esophagus and other structures they grow, they lengthen up, it ultimately comes to lie in the lower chest or upper abdominal region, where it will form the diaphragm. Now what actually happens that somewhat here there will be the heart, and we will discuss those details in the upcoming lecture, that somewhat here there would be the heart. So this part of intraembryonic celom, it is going to form the pericardial cavity, and this part is going to form the peritoneal cavity. And here this part, it is also going to form the pleural cavity. Why? Because here from this primitive GI tube, the tracheoesophageal diverticulum will arise, and from this diverticulum the lung bud will form and lungs will be formed. So it will form the pericardial cavity as well as the upper part of pleural cavity. And what about the lower part of pleural cavity? It is formed by another canal. So let me show you exactly what I mean by this. Now here we can see actually this intraembryonic coelom, it has been divided into two parts, three parts actually. One part is this primitive pericardial cavity, the other part is primitive peritoneal cavity, and the third part is basically these two canals behind the septum transversum. So here is the septum transversum, and behind the septum transversum, lateral to primitive GI tube and the dorsal mesentery, lateral to these structures, here this canal, let me show you exactly. Here is this canal behind the behind this septum transversum and lateral to the primitive GI tube. This canal is connecting the primitive pericardium to primitive peritoneum. So this canal is called Pericardioperitoneal canal. So right pericardioperitoneal canal, and on the other side there will be the left pericardioperitoneal canal. And please remember, this pericardioperitoneal canal, it is lateral to the primitive GI tube as well as the dorsal mesentery, and it is behind the septum transversum. So what we can say that the intraembryonic coelom, it has been divided into three parts: the primitive pericardial cavity, which will also form a part of pleural cavity, but we just right now we will just call it primitive pericardial cavity. So primitive pericardial cavity, primitive peritoneal cavity, and then there are these two canals. Here it will be the left canal and here will be the right canal. If we go deeper, here will be the right canal, right peritoneal canal behind the septum transversum. So please remember these structures, and we will talk more about these structures and how these structures form the pleura, pericardium and peritoneum, that we will discuss in the next lecture. Finally, I would like to briefly talk about the origin of septum transversum, because it is quite confusingly presented in the embryology textbooks. Actually, there are two hypotheses for the origin of septum transversum. So let's first talk about the hypothesis number one, and that is, as the small spaces appear within the intraembryonic mesoderm and these spaces coalesce, actually no such spaces form at the cranial end, and resultantly what will happen that a block of tissue will remain at the cranial end like this. So here you can see that at the cranial end this block of tissue remains, and this block of tissue is the septum transversum. So according to this hypothesis, there is no particular, we cannot say with certainty that whether this septum transversum, it arises from the intraembryonic somatic mesoderm or if it arises from the intraembryonic subplanchnic mesoderm. Actually, we can say that it develops from the unseparated part of intraembryonic mesoderm. It develops from the cranial end of intraembryonic mesoderm, where the intraembryonic mesoderm is not split into the somatic and subplanchnic layer. So at the cranial end, if the intraembryonic mesoderm is not split into subplanchnic and somatic layer, this block of tissue is formed, and that is the septum transversum. Then there is another hypothesis of its origin. So according to other hypotheses, actually the intraembryonic coelom completely forms, and here that whole this structure is completely split into the intraembryonic somatic layer and subplanchnic layer, but from the intraembryonic subplanchnic layer a block of tissue later originates like this, and this block of tissue is actually the septum transversum. So according to the second hypothesis, the septum transversum arises from the intraembryonic subplanchnic mesoderm, but according to the first hypothesis, we do not know precisely. Actually, we cannot say precisely that from which mesoderm the septum transversum arises, because it is not even split. So it arises from the cranial unsplit part of intraembryonic mesoderm. And another thing that I would like to talk about, that according to some authors, at the cranial end, this U-shaped intraembryonic coelom, it is not continuous with the extraembryonic coelom. The septum transversum is still there, but here there is that curtain-like separation between the intra and extra embryonic celom. I don't really know what is the significance of this separation, because many textbooks, they don't simply mention or they don't simply show in the illustration this separation. So I don't really know that what is the significance of this, but because it is mentioned by some well-known authors, so I thought that it is worth mentioning here. So that was about the body cavities, and in next lecture we will talk about the formation of pericardium, pleura and peritoneum. Thank you so much for watching this video. Have a nice day.